Hello everyone, welcome to AS and A-Level Biology with Dr. Demi. I am Dr. Demi, and in today's video I will be sharing with you Chapter 2 of your AS Biology syllabus, which is Biological Molecules. So if you have simply stumbled on my channel for the very first time, I suggest you hit the subscribe button so that you can get notifications every time I post a new video. Also, I want to make it clear that I am posting the AS and A-Level Biology syllabus in chronological order, so you can use this channel for your revisions. If you're just starting out your AS biology journey, then this is definitely a good idea for you. If you're revising for the exams next year for June or October, November, then this is also a good idea for you. So don't forget to hit the subscribe button and also invite your friends to join in as well. So today we are going to be starting with the second chapter, which is biological molecules. Now, when you think of biological molecules, some people said in my class when I asked them that they were thinking about things that were very chemical in nature and not necessarily things that they could relate to. If you look at the syllabus, you will see that you are expected to know 4 different biological molecules, or 4 classes of molecules if you want to look at it that way. Those include the carbohydrates, proteins, as well as the fats and lipids and water. So we're going to start with the carbohydrates today, and this is a fun fact that I usually share with people, that usually when you think of carbohydrates, you are told that they are for energy release. As a matter of fact, people say that if you don't eat enough carbohydrates, then your brain would not get enough energy and it would have to rely on lipids, which is then what puts you in a ketosis. So a ketosis is what, for example, the ketogenic diet is based on. So when you don't eat enough carbs, you don't have enough energy from carbohydrates, so your body starts to rely on fat. But the fun fact here is that even though carbohydrates are associated with providing energy, eating refined carbohydrates, especially in large amounts, can make you tired. So when you eat a lot of pasta, you eat too much bread, you basically eat too much of refined stuff, you find that you want to sleep. You just want to lie down a little bit because you're exhausted. That is a fun fact about carbs. So the fact that it's a carbohydrate doesn't necessarily mean that it will give you energy. In some instances, it can make you tired. Okay, enough of fun facts. Let's get into the main study. So the first thing you have to do is to understand the basics of carbohydrates. Carbohydrates are made of monomers and polymers, right? So when we say a monomer, we are simply speaking of a single block of carbohydrate. So over here, that is why I have the picture of a brick over here. It's not to stone anyone, it's just to let you know that a single brick such as this would be considered a monomer. So monomers are simple building blocks that are joined together to make a polymer. And so if we want to imagine it that way, we can simply then say that a polymer would be a brick wall such as this, because it is the joining of many monomers together. So a polymer is a giant molecule made from repeating subunits of monomers. So think of it again, the monomer is the single building block, while a polymer is many of monomers joined together in order to make a giant molecule. This here is a schematic that I used to help my students remember the different monomers for the polymers they have to learn. So as you can see, I have said here, remember foam. Foam simply stands for fatty acids, organic bases, amino acids, and monosaccharides. Of course, that is not necessarily exhaustive, but the idea is that if you can remember the very first part of the monomer, you would be able to remember what follows with it. So for example, in order to make a lipid, we have to join fatty acids with glycerol. We will look at that in detail when we go into lipids. We have to join organic acids and nucleotides in order to make nucleic acids. We have to join many repeating chains of amino acids together in order to make proteins, and we have to join many monosaccharides together to make a polysaccharide. So if we're going to use the building block example, then we can say that the single blocks are things like your amino acids, your monosaccharides, your organic bases and nucleotides, or your fatty acids and glycerol, while the big wall, the big brick walls, would be the lipids, the nucleic acids, the proteins, and the polysaccharides. Just to clarify here, polysaccharides are carbohydrates, but I will go into detail as we go on. So let's get into carbohydrates. First question is, how was the name carbohydrate formed? This is important because it helps you break it down and help you just understand in detail why carbohydrates are so called, and also helps you understand their structure. So the first thing you have to bear in mind is that carbohydrates contain carbon, hydrogen and oxygen, and the carbo part refers to the presence of carbon, while the hydrate part refers to the presence of hydrogen and oxygen. Obviously, you should know that when you combine hydrogen and oxygen together, you're likely to get water, which is what you will see in the general formula of a carbohydrate. You can see there that it is CxH2OY, and what the X value indicates is basically the number of molecules of carbon, and the Y value indicates the number of molecules of hydrogen and of oxygen. As you can see, the number of molecules of hydrogen is double that of oxygen, which is why whatever hydrogen is, whatever oxygen is rather, you have to multiply by 2 to get the number for hydrogen. But you don't have to worry yourself too much about that. I feel like I'm going into a little bit of chemistry, but you will be able to see as we go along what the values look like. So again, just reinforcing what we have already mentioned, that carbohydrates can be split into monosaccharides, which would be the monomer. You can have disaccharides. Disaccharides simply indicates that there are two monomers that have been joined together. So two monosaccharides have been joined together to make a disaccharide. And you can have polysaccharides, which indicate that many monomers have been joined together in order to make a polysaccharide. And again, I have used the bricks here to indicate that, so that you can see that a monosaccharide is simply a single molecule, a disaccharide is two molecules joined together, while a polysaccharide is many of the monosaccharides joined together. This image also contains some of the examples of monosaccharides. So for example, you already know about glucose. It is contained in many of our carbohydrate foods. It is a monosaccharide and it is necessary for us to have energy. We also have fructose, which is the sugar that is found in fruits. We have galactose as well. And what you will see here is that all of these are sugars, which is why their names end with OSE. So wherever you find the OSE ending, then the molecule you're dealing with is most likely a sugar. The disaccharide, as you can see, is either maltose, sucrose, or you can have lactose, which is what is found in milk. And you have your polysaccharides such as starch, glycogen, and cellulose. We will go into detail to see how these function in the body. So again, just reinforcing the idea of monosaccharides, you can see that there are different types of monosaccharides themselves. You can have the trioses, which indicate that you're dealing with a 3-carbon sugar simply by looking at the prefix tri, T-R-I. That tells you that this is a 3-carbon sugar. A pentose, if you think of the fact that a pentagon in math has 5 sides, then you see that a pentose is referring to a 5-carbon sugar. While a hexagon has 6 sides, then that is a 6-carbon sugar. In this case, you will mostly be dealing with hexoses, but when we go along, you will find that we encounter some pentose sugars, especially when we are discussing things like the DNA, and you will see that we encounter triose sugars when we discuss respiration. So examples of these, the triose, you have glyceraldehyde, hydroxyacetone. For pentoses, you have ribose and deoxyribose. Ribose is the sugar that is in RNA, and deoxyribose is the sugar in DNA, which carries all of our genetic information. You also have the excesses such as glucose, fructose, and galactose. These are 6-carbon sugars, and all of these, irrespective of the number of carbons they have, are monosaccharides, which means they are single building blocks for other types of carbohydrates. So let's zoom into glucose, which is our most important monosaccharide for this chapter. If you look at the formula for glucose, you will see that it is C6H12O6. What this means is that glucose has 6 carbons, 12 hydrogens, and 6 oxygens. Now, if you look on the far right, you will see that there is a structural formula for glucose where you can count 6 carbons, you can count 12 hydrogens, and 6 oxygens. And that would be great right, if this is the way glucose was always represented. But what you're most likely to find is the structure on the far right, on the far left rather, which is a ring structure of glucose. I advise students to learn how to draw the structure, including the exact arrangement of the hydroxyl and hydrogen compounds, or hydrogen ions rather, so that you are able to identify the different types of glucose, which is something you will see on the next slide. So if you look at this structure, obviously the first thing that comes to mind is, well, where are the carbons over here? Because you can only see the hydrogens and the oxygens and maybe one carbon up there. Well, the carbons are simply indicated here where the lines are meeting. So I have drawn arrows to show you the different carbons, and if you see, you can count about 5 carbons over there, and the 6th carbon is written up there as CH2OH. Now, something that I always ask students is, how many types of glucose are there and how can you differentiate between them? There are 2 types of glucose molecules. You have alpha glucose and beta glucose, and the difference between these two is the position of the hydroxyl group on carbon number 1. And of course you might say to yourself, well, how do I know which one is carbon number 1? This is why I have shown you how the carbons are numbered. So we usually start from the carbon on the very far right of the ring structure, that is carbon number 1, and we label in a clockwise direction, which means carbon number 1 is the very first one, is the one on the very far right, and the one beneath it is carbon number 2 and 3, 4, 5, and carbon number 6 is the one labeled as CH2OH on the molecule. Now if you look at the hydroxyl group over here, you will see that on the alpha glucose you have the hydroxyl group on the bottom of carbon number 1, while with the beta glucose you have the hydroxyl group on the top. I tell students an easy way to remember this is to simply bear in mind that the alpha is always on top except when it's in glucose. So if you remember that, you will then know that with the alpha glucose, the hydroxyl group is always at the bottom. This is a very important part of this chapter because I have seen lots of questions where you are asked to identify what kind of glucose you are looking at. And also again, I just want to reiterate that it is important for you to be able to draw the different structures of glucose, that is the alpha glucose and the beta glucose, because it is a very popular paper 1 question to draw very confusing structures and ask you to identify which one is or is not glucose. Now these are scientifically called isomers simply because they are mirror images of each other due to that switch in the position of the hydroxyl group in alpha and beta glucose. So on this slide, I am simply showing you the most common disaccharides. So we have maltose, sucrose, and lactose. Maltose is the addition of glucose and glucose, which means 2 glucose monosaccharides forming maltose. You have sucrose, which is glucose plus fructose. Fructose is the sugar that's found in fruits, and you have lactose, which is the sugar found in milk, and it is made from glucose and galactose. On this image over here, I have shown you the addition of glucose and fructose, and one thing that you will notice about fructose is that it has a different structure from glucose, but it is also a 6 carbon sugar. So it is important for you, I would advise, to just pause here and have a look at the structure of fructose. I have also seen questions where you are being asked to identify which one is fructose, or you are being asked if fructose is truly glucose. And so I just want to point out that just because it has 6 carbons doesn't mean that it is glucose. You need to look at the arrangement. So fructose over here looks a bit like a house if you're drawing a stick structure of a house, while glucose is more of a hexagon. So how do disaccharides form? Well, you can have 2 glucose units as we have in this image over here. And what you will notice immediately is that the hydroxyl group on both glucose units have been highlighted in red, Why this is important is because those hydroxyl groups will react with each other to form water and what we call a glycosidic bond. So if you look on the right side of this chemical reaction, you will see that there is an oxygen there that is being pointed to as the glycosidic linkage. That is what we call the glycosidic bond. And we have water that is being released from the formation of this bond. If you want to write it out in simple terms, you can simply say the OH from one carbon plus the OH from another carbon will give you water, which is H2O, and an additional oxygen. The H2O is given off as a waste product, while the oxygen is used to form the bond, which is called the glycosidic bond. Something to bear in mind with carbohydrates is that they always have glycosidic bonds. This will become important as we start to learn about the other biological molecules, and you will learn about new bonds that they form. It is also important to bear in mind that this is a condensation reaction. So whenever we have the joining together of two molecules, which then results in the release of water, we call that a condensation reaction. So condensation is joining together of two molecules and releasing water as a byproduct. And in the process forming a bond, like you can see here, we form the glycosidic bond. The opposite of a condensation reaction is hydrolysis. Hydrolysis means the opposite. So we are adding water into a molecule in order to break it apart. So if, for example, we were to pick maltose, which is the addition of glucose and glucose, it means that if we want to break down maltose, we have to put it through a hydrolysis reaction by adding water to it in order to get our two molecules of glucose. And this is a polysaccharide, again just reinforcing that polysaccharides are made from many repeating units of a monomer, in this case of a monosaccharide, and they have many glycosidic bonds, which means that to form a polysaccharide you have to undergo many condensation reactions and release molecules of water. Polysaccharides are not sugars, and this is something that needs to be reinforced in students. They can be broken down into sugars, but when you combine a lot of sugars such as glucose, for example, to make glycogen or to make amylose or amylopectin, which are molecules you will learn about, then you're no longer going to have a sugar. What you will have is either a storage molecule or a structural molecule. So the two types of polysaccharides are either for storage or for structure. So for example, you have starch, which is a storage molecule in plants, and you have glycogen, which is a storage molecule in animals. For structure, you have cellulose. Cellulose is a structural molecule which you can find in the cell wall of plants. So if you look to chapter 1, you will remember that we discussed the cell wall and we said it is for rigidity and support. It is actually made of cellulose fibers. And you also have a molecule called chitin or chitin, depending on how you pronounce it, and this molecule is also for structure, and you will likely find it, for example, in your prawn shells. So if you're a person who loves seafood, when you peel off the shells of the prawns, that shell contains a lot of chitin. The most important polysaccharides you would have to learn are starch, glycogen, and cellulose. You don't have to know chitin. Now, the reason why we make polysaccharides is because we cannot store glucose in its natural state within our bodies. This is because when we store glucose, glucose is a very reactive compound and it can easily interfere with some of the processes within the cells. Besides that, storing glucose inside the cell can make the inside very concentrated and then affect the osmotic potential of the body, as you will see when we get to chapter 4. So what happens then is that plants, like I said, will store glucose in the form of starch, whereas animals will store glucose in the form of glycogen. This is very important because it plays a role in whether or not a person can develop issues like diabetes. If you're unable to easily convert glucose into glycogen, then you might have a diabetic condition, but we're not going to go into that. That's a chapter 14 information. Now, starch, like we said, is the storage molecule in plants. So when you eat a potato, for example, you are eating the starch that has been stored within the plant. And when we get to chapter 7, you will see how this is formed. So starch itself is not just made directly of glucose. It is made of amylose and amylopectin. These are two different compounds that are made from glucose. So if you want to think about it, you have amylose and amylopectin, which are made from glucose, and then they come together in order to form starch. I'm going to go through amylose very quickly and then go into amylopectin. If you look at amylose on this slide over here, you will see that it is a straight chain compound. And when you look at the bonds between it, you can see there that they are glycosidic bonds as expected, because you are simply forming polymers of glucose. Now, something to bear in mind is that amylose has what we call alpha 1,4 glycosidic bonds. When we say something is alpha 1,4, first thing you should bear in mind is that that alpha simply means that amylose is formed from alpha glucose. Okay, amylose comes from alpha glucose. When we say it has 1,4 glycosidic bonds, it means that the glycosidic bonds are formed between carbon number 1 of one glucose molecule and carbon number 4 of another glucose molecule. So I will encourage you to actually look at this image. You can pause the video here and try to count the carbons using the technique I showed you earlier, and say to yourself, well, where is carbon number 1 and where is carbon number 4, so that you can see that this is so. So between carbon number 1 and carbon number 4 of every molecule of glucose in amylose, you have a glycosidic bond, and that is why we call it a 1,4 glycosidic bond. It is also important to remember that amylose is formed from alpha glucose. When you look at amylopectin, on the other hand, amylopectin has what we call the alpha 1,6 glycosidic bonds as well as the alpha 1,4 glycosidic bonds. So you can think of amylopectin as structures of amylose that are then joined to structures that are above or beneath them in order to form a branched chain. So alpha 1,6 glycosidic bonds indicate that the bond is formed between carbon number 1 and carbon number 6 of another glucose molecule, and again they are made from alpha glucose subunits. I will encourage you again to pause the video, have a look at it, and count the carbons in order to be sure that this is so. Starch grains are found in your chloroplast in plants and in foods such as your potato tubers, your cereals, and even your legumes. This is something interesting that my students just couldn't believe, the fact that you also have starch in foods like beans, because they always thought that beans were purely protein. So that was just something interesting and might also be a fun fact for you. In animals, we don't store starch. We actually only store glycogen. And the only thing you have to know about glycogen is that it is very similar to amylopectin, which means that just like amylopectin, it has alpha 1,4 glycosidic bonds as well as alpha 1,6 glycosidic bonds. But the difference between glycogen and amylopectin is that glycogen has more branches. So that means it has more alpha 1,6 glycosidic bonds. But the structure is essentially the same. So remember that glycogen is made from alpha 1,4 and alpha 1,6 glycosidic bonds. It is similar to amylopectin. Starch is made from amylopectin and amylose. Amylose has alpha 1,4 glycosidic bonds and amylopectin has alpha 1,6 glycosidic bonds. Breaking down starch and glycogen to release energy is controlled by enzymes, and that is something we will go into detail in when we go further in the syllabus. The last polysaccharide that you have to know is cellulose. Cellulose has a structural function because it is mechanically strong. So if you have been following trends such as the production of bioethanol, you will learn that there have been experiments to convert cellulose from plant barks or from waste agricultural resources into ethanol, and that is proven difficult, or it used to prove difficult rather, because of the presence of cellulose. Cellulose is present in the the cell wall of plants, and it is made of beta glucose. So unlike glycogen and starch, which are made from alpha glucose, cellulose is made up of beta glucose. It has a very high tensile strength that is almost equal to that of steel, and cellulose fibers are permeable and they allow water and solutes to enter and leave the cell membrane. So that is it from me today. I hope that you have been able to get some revision in by watching this video. If you have any questions, please post them in the comment section and I promise to answer them as soon as possible. I will be posting more information on biological molecules. Like I said, this chapter is dedicated, this channel rather, is dedicated to the chronological content of the AS and A-level biology syllabus, and I hope that it is helpful to you as you prepare for your exams, or even as you just start your AS and A-level biology journey. Thank you so much for watching. Don't forget to hit the subscribe button. Until next time, have a good day. Bye.